Hello viewers, today I am going to deliver the lecture on why zebrafish make an excellent research model. Myself I am Pandian, working as a tutor in Physiology Department, D.Y. Patil Medical College, Kolhapur. Why the zebrafish make an excellent major role in this research, we will see in this particular lecture. Let me start. This topic going to give a brief introduction about zebrafish, why it is play a major role in research method. That result is it's give an excellent result in the particular research work which is we have going to do. Why using animal in research? It is an important role in minimizing animal suffering by using the least sentient organism possible to answer the question at end. But there are also purely scientific reasons why lower animals are, such as zebrafish can create a better model system. Some of these reasons are outlined in the following slides. Contents. In this study we are going to discuss about this entire contents and subtitles. First one is introduction. In 1960s, zebrafish started using as a model organism in research work. The size of the fish is 2.5 to 4 centimeter in length. In larval state it is transparent, and matured adult fish has a blue color stripes from head to tail, which is denoted their breed as well as development. The complete genome sequence of the giraffes was published in 2013. His genomes are around 1 million base pair in length and contains around 26,000 protein codes genes. It is the actual pictures of zebrafish. We can see now from head to tail that is a blue stripe which is denoted as breed as well as the adult or embryo level of the zebrafish. He is the first person innovated the particular zebrafish model organism from University of Oregon, USA by George in 1970. He is the founder father of zebrafish developmental and genetic research. What is the benefits of zebrafish? Why we have chosen this in this particular research work nowadays? The zebrafish is small and very robust, and they are cheaper to maintain than mice. And some fishes are particularly when you're taking that breed and collecting the eggs, it's only take over night hours. In the zebrafish, you can take everything in day hours. It is a main advantage for research workers because they can able to sleep in the overnight. They can do all the research studies or methodology in day hours. And zebrafish produce hundreds of offspring at weekly intervals, providing a scientist with ample supply to embryology or embryological studies over there. It is a one more advantage to choosing the particular zebrafish in the research work because it gives us a hundred of offspring is compared with the mice or mammals we are have to wait for at least 3 weeks. They grow at extremely fast rate, developing as much in the day as a human embryo develops in 1 month. You can see here from zygote to the 48 hours are the embryos. From the development, each and everything is transparent. Under low power microscope, we can check entire clinical features, anatomical features, and genetic related to the disorders. Everything we can check in the particular stages with low power microscope. Zebrafish embryos are nearly transparent, which allows researchers to easily examine the development of internal structures of particular offspring. Every blood vessels in living zebrafish embryo can be seen using just a low power microscope. Already I said, as zebrafish eggs are fertilized and develop outside the mother's body, it is the ideal model organism for studying early development. Here I am showing comparative study of human as well as zebrafish. Why we have chosen it? Zebrafish have a similar genetic structure of human. They share 70% of genes with us. 80% of gene known to be associated with human disease has zebrafish counterpart. Here, as a vertebrate, the zebrafish has the same major organ and tissue as the humans. Their muscle, blood, kidney, and eyes are many features with human system is correlate with the particular fish. I can give a one example here. That particular heart is get regenerated by their own without any medicine or what some other reasons. Here uninjured heart is there in the section A, B, C, D, E. From when you cut the particular ventricle, you can see it is own it get cluttered and it is get regenerated, and then remodeling is take over by their own fish. In human being it is not take over. Once any injuries take over, we will get the scar only. For good example, zebrafish have a unique ability to repair their heart muscle. For example, if the part of the heart is removed, they can grow it back in matter of week. Scientists are working to find out the specific factor involved in this process to see if this will help us to develop way to repairing of our human heart with heart failure or who have suffered heart attacks. The zebrafish genome has been fully sequenced to very high quality. This has enabled scientists to create a mutation in more than around 14,000 genes to study their functions in the human heart diseases. During heart attack, heart muscle cells getting more very less oxygen supply to the particular tissue level, so it's become die and it is leave the scar tissue. Scientists are working to find out the specific factor involved in this process to see if this will help us to develop our way of repairing the heart in the human with heart failure or who have suffered heart attacks. Already I told there is a particular uh, achieved process for regenerative, that is SMAT3 and dependent TGF beta signaling. These two processor signaling is make us get an idea from the zebrafish how it is regeneration processes take over. Genetic similarity to the human. Zebrafish have a particular characteristic, that is a high degree of sequence and functional homology with mammals including human beings. Due to that conservation of the cell biology and developmental process across the vertebrates, studies in fish can give a great insight into the human disease process. For a good example, to get the protein study, particularly gene sequencing and gene signaling and particular secondary signaling processes. It is the best easy to handle it as well as compare with mammals due to their small size and the relatively simple nature of their natural environment. It is easier to keep the zebrafish in more natural condition than it is possible to stimulate for mammals. This minimize housing stress and the impacts such as the stress may have on outcome of the experiments. For mammals, we have to spend more time to keep it them in the environmental side as well as the stress factors, and we can't able to monitor entire hours. But here it is not like this. Once you have to fix this temperature, it can maintain their own temperature by their own blood circulation and all, so you can never bother about it. It is a easy way to do the research work in zebrafish. Impact of any genetic mutation or drug treatment, it's very easy to identify the factors which is involved in the embryological level as well as offspring level. Zebrafish embryos and larvae are completely transparent, meaning that it is possible to follow the impact of genetic manipulation or pharmaceutical treatment using non-invasive image techniques. Less invasive techniques minimize animal suffering. The invasive procedures avoided not only impact on the welfare but may also affect the experimental outcome. So the transparent nature of the zebrafish larvae could also mean results are more accurate and easier to reproduce. Lots of offspring. In this particular, ensuring the readily supply the animal for research is also easier with the zebrafish. Zebrafish have a much larger number of offspring in each generation than rodents. Rodents have a 5 to 10 offspring per pairing and comparison to the 200 to 300 obtained from zebrafish. Here if you want to take a genetic level studies for mammals, you can get only 3 or 4 offspring. In that also something will never grow up, we can't able to see the results. But here it is not like this. From the zygote to the maturation, that is 48 hours, you can check entire processes, mutations, anatomical study, genetical study, physiological and morphology of the particular organs we can study in the zebrafish. It is a one more advantage why we have to choose the zebrafish here. Easier to introduce genetic changes. Zebrafish embryos are able to absorb the chemical that have been added to their water, meaning it easy to introduce changes to their genes using nothing more than chemical mutagens. Zebrafish are able to withstand much higher level of chemical mutagens then can be tolerated by rodents or mammals. So it is possible to induce a much higher intensity mutation in their genomes. For good example is now the studies are going on in the Western country as well as in India in few places, that is cancer studies. Zebrafish have been used to help the unlock number of biological process behind the muscular dystrophy and are the important model for understanding the mechanism of development and disease, particularly cancer. So here you can comparatively you can see upper one is the normal one, the below one is cancer induced zebrafish. How they have taken the cancer cells from the human. It is a picture shown that we want to collect the sample from the human being. We want to induce into the particular tumor cells to the zebrafish by the way of injection, and then we want to wait for getting migration, proliferation, and other processes. Then finally, you can take the particular content of the zebrafish or embryo or adult one, and we can go to the particular drug activity or sequencing. It is a main useful cases for developing and signaling process for cancer institutes. How to do the benefits for using the zebrafish? Despite the growing interest in the biodata approaches to study the genetic of the human disease, currently there is a still need to work more in the animal model system. For many zebrafish are becoming a increasingly popular choice of model. Biomedical research are taking advantage for the vast amount of genetic information available in the public database and medical records to explore the causes of human disease and gain insight to help the development of the novel as well as personalized treatment. But this entire approaches does not have a limitation. Mainly, particularly for zebrafish, we have a limitation for everything, because when you're giving the more drug or lethal dose, if you are increasing furthermore, it can withstand, but you can't able to get the results because there is a lethal index of the zebrafish is very less compared with the mammals. That is the only one drawback why we are not taking the zebrafish nowadays. Is the other uses zebrafish as a model is used to study. Particularly nowadays, very many cases are there for heart regeneration. Many lectures they are available in the online, and tail regeneration. Particularly, it is useful for who have semi-paralysis and hemi-paralysis persons. Retinal regeneration, these studies are going on in diabetology. Human pigmentation, it is very well known. Cosmetic research work is going on, and then in India, it is going on cancer research work in ICMR as well as AIMS. And autism, it is take over all over the world in the neurologist rated studies and etc. Those things are available where I have taken entire thing. It is a reference I have mentioned here. Thank you so much for watching this video, and please do search in that particular content in the zebrafish. Thank you so much.